A negative mind will never give you a positive life. Believe in yourself. Whoever is trying to bring you down is already below you. I'm Chitra, welcoming you all to this online class. Students, we have finished 4 chapters and today we will start a new 5th chapter, Molecular Genetics. Mendel's theory dispelled the mystery of why traits seem to appear and disappear magically from one generation to the next. Mendel's works reveals the patterns of heredity and reflect the transmission of evolved information from parents to offsprings. This information is located on the chromosomes. One of the most advanced realization of human knowledge was that our unique characteristics are encoded within a molecule of DNA. The discovery that DNA is the genetic material left several questions unanswered. How is the information in DNA used? Scientists now know that DNA directs the construction of proteins. Proteins determines the shape of the cell and the rate of chemical reactions such as those that occur during metabolism and photosynthesis. The hereditary nature of every living organism is defined by its genome, which consists of a long sequence of nucleic acids that provide the information needed to construct the organism. The genome contains the complete set of hereditary information for any organism. The genome may be divided into a number of different nucleic acid molecules. Each of the nucleic acid molecule may contain large number of genes. Each gene is a sequence within the nucleic acid that represents a single protein. In this chapter, we will discuss about gene as the functional unit of inheritance and which is the genetic material, whether it is a DNA or it is a protein. DNA is the genetic material with several experiments and the chemistry of nucleic acids and RNA world. What is a gene? Gene is a basic physical and functional unit of heredity. Genes as the functional unit of inheritance. First, we see what is genetics. Genetics is the study of transmission of hereditary characters from one generation to the other generation. So, the characters are transmitted from the parent to the offsprings. That transmission is called as genetics. Gregor Johann Mendel, he is called as the father of genetics because he conducted the experiments for this transmission of characters. So with help of the pea plant, he did so many experiments. The concept of gene was first explained by Gregor Mendel in the year 1860. So Gregor Mendel explained what is gene, how it carries the hereditary characters from the parent to the offsprings with help of a pea plant experiment, and how these characters are formed, how these characters are transformed. So he called these characters as factors. He called it as factors. He never used the term gene. Mendel factors are now called as genes. Wilhelm Johansen coined the term gene in the year 1909. He is a Danish biologist. A gene is a physical and functional unit of heredity. Number of genes in an organism is more than the number of chromosomes. Hence, several genes are located on the same chromosome. According to Sutton, in the year 1902, the genes are arranged linearly in a chromosomes. Genes are present in chromosomes. Chromosomes are present in nucleus in the cell. The genes are not scattered in the chromosome, but they are arranged linearly, orderly, or in a straight line in a chromosome. You can see in this picture, the genes are arranged, that red colored part, no? So these genes are arranged in between the non-coding DNA. So the genes are arranged linearly in a chromosomes. It is a main important property of the gene. So it looks like beads on a string, and each gene occupies a specific position on the chromosome. If you see the properties of genes, genes exist in alternative forms called alleles. Alleles is nothing but the alternative form of a gene. You can see the example tall and dwarf. Genes undergo sudden changes, that is called as mutation. Sudden changes takes place in the position and composition of the genes. So these mutant genes will cause several genetic diseases. Genetic and hereditary diseases are caused by sudden changes in the position and composition of a gene. Gene can undergo self-duplication. That means they are capable of producing their own copies of genes. Beadle and Totem in the early 1940s proposed a hypothesis called one gene, one enzyme hypothesis, which states that one gene controls the production of one enzyme. So, this work can be carried out on Neurospora carasa, that is a red bread mold. Mold is nothing but a fungus. So you can able to see fungus in the breads. So green color and red color fungus will form on the bread. So it states one gene controls the production of one enzyme. So it is carried out in red bread mold, that is Neurospora carasa. Hofminister in 1848 discovered that during mitosis, that is a type of cell division, the nucleus organizes itself into rod-like structures called chromosomes. So before cell division, the chromosomes are thread-like structures inside the nucleus. But at the time of cell division, they organize themselves into a rod-like structures. Miescher in 1869 isolated substance from the nuclei, and he called it as a nuclein. So nuclein is a substance isolated from nuclei, and it was renamed as nucleic acid by Altman in the year 1889. Nucleic acid are of two types, which is now known as DNA and RNA. Many experiments were conducted out to study the actual carriers of genetic informations. However, chromosomes are eukaryotes containing several proteins along with DNA, that is deoxyribonucleic acid. Scientists could exactly confirm whether genetic material in the chromosome is DNA or proteins or both. By around 1926, research for mechanism of hereditary material had reached the molecular level. Yet, the genetic material had still not been discovered. In 1928, Frederick Griffith, a British scientist, conducted an experiment and accidentally demonstrated the transformation of genetic material in bacteria. In fact, he was in process of developing vaccine against bacteria Streptococcus pneumoniae that causes pneumonia by inoculating the dead microbes, along similar lines of Louis Pasteur experiments on vaccination. Frederick Griffith grew bacteria in a cultured plate and found that they would have two forms, the smooth and the rough. This was because the smooth shining colonies on the S strain of bacteria possess the mucus on the polysaccharide coat, while rough colonies of R strain do not possess saccharide coat. Further, when the mice were injected with S strain, they died in pneumonia infection, while those injected with R strain did not develop pneumonia. This proves that S strain of bacteria is virulent and R strain is harmless. However, Frederick also found that heating killed bacteria. So, when he injected the heat killed S strain bacteria to the mice, they did not die. On the other hand, when he injected the mix of R strain bacteria and heat killed S strain bacteria, the mice died. Surprisingly, he even recovered the living S strain of bacteria from dead mice. So, on the basis of this experiment, he concluded that the heat killed S strain bacteria has somehow transformed the R strain bacteria. He also concluded the reason why the R strain bacteria produce the smooth saccharide coat and became lethal and virulent because of transfer of some genetic material from the heat killed S strain bacteria. However, there was still no means to identify the genetic material that was transferred and responsible for transformation process. Later, after 16 years in 1944, three scientists, Oswald Avery, Colin McLeod, Macklin McCarthy, once again began the experiment conducted by Frederick. At that time, they tried to identify the biochemical nature of the transforming principle of Frederick experiment. For this, the scientists first purified the biochemicals, proteins, DNA, and RNA from the heat-killed S-strain bacteria. They did so to identify the biochemical responsible for transforming the live R strain into S strain bacterial cell. Through their experiment, it was discovered that it was DNA from S strain bacteria that had transformed the R strain bacteria. They also found that digestion of DNA of heat killed S strain with DNAs in evident transformation. Further, the scientists also discovered that digestion of proteases or protein digesting enzymes and RNases or RNA digesting enzyme did not affect the transformation. This led to the conclusion that proteins or DNA will not a transferring substance and implied that DNA was the cause for transformation. Thus, Oswald Avery, Colin McLeod, Macklin McCarthy concluded that DNA is the heredity material in most of the living organisms. Nevertheless, many biologists has unconvinced that DNA is the genetic material, and so the research was continued for several years and was concluded with the discovery of DNA. In 1953, Alfred Hershey and Martha Chase confirmed DNA by demonstrating DNA is the genetic material of virus called phage T2. The phage which infects the E. coli consists of head, sheath, tail, and base plate made up of different proteins. DNA is packed within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacteria by its tail. Next, the phage injects the genetic material into the cell. The genetic material directs the bacterial enzyme to produce viral offerings. When life cycle is complete, 100 to 200 progeny phage is inside the bacteria. When the bacteria breaks or lyses, the phages are released. Since the T2 consists of only DNA and protein, the Hershey and Chase reasoned that the genetic material must be one of the two components. So, they designed an experiment to find which it is. For this experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive protein. To make the components radioactive, Hershey and Chase infected the E. coli with T2 and grew the bacterial cells in two different culture media. one containing the radioactive isotope phosphorus 32 and the other containing the radioactive isotope sulfur 35. All the phages released from the bacteria grown in phosphorus 32 medium had radio-labeled DNA, while all the phages released from the bacteria grown in sulfur 35 medium had radioactively labeled proteins. In the next phase of the experiment, Hershey and Chase infected E. coli with two types of labeled T2. Then they used a blender and centrifuge to separate the protein parts of the phage called phage host from the de-infected cells of virus. The scientists discovered that the labeled DNA appeared in the host cell but not in the phage host, while the labeled proteins appeared in the phage host, not in the host cell. In addition, the progeny of the T2 with the labeled DNA will radioactive, while the progeny of the T2 with labeled protein is not radioactive because genetic material is passed on from parent to offerings. Hershey and Chase concluded that DNA had to be the genetic material of the T2. The results has convinced the scientist community that DNA was the genetic material or the heredity material. Next, we move on to the chemistry of nucleic acids. That means already we identified the genetic material is DNA, and we have to find out the chemical structure of these molecules. So generally nucleic acids are a long chain or a polymer of repeating subunits called nucleotide. That is, nucleic acids are made up of nucleotide, and each nucleotide has subunits, and its subunits has three parts: nitrogenous base, a pentose sugar, that means a 5-carbon sugar, a phosphate group. First is the pentose sugar. There are two types of nucleic acid depending upon the types of pentose sugar. They are DNA and RNA, that is deoxyribose sugar and ribose sugar. Ribose sugar is called as ribonucleic acid and deoxyribose sugar is called as deoxyribonucleic acid. So this DNA is found in the nucleus of eukaryotes and nucleoid of prokaryotes. So the only difference between these two sugars is that there is one oxygen atom less in deoxyribose. In deoxyribose, only one oxygen atom is less. This is the difference between the DNA and RNA. Next, we move on to the nitrogenous bases. So what are nitrogenous bases? The bases are nitrogen containing molecules having the chemical properties of a base. What is a base? Base is a substance that accepts the hydrogen ion or a proton in a solution. So this DNA and RNA both have 4 bases, 2 purines and 2 pyrimidines in the nucleotide chain. 2 of the bases, adenine and guanine, have double carbon nitrogen ring structures and are called as purines. The bases thymine and cytosine in DNA have single ring structure and these are called as pyrimidines. But in RNA, thymine is paired with uracil. So cytosine is replaced by uracil in RNA. Thymine is unique for DNA while uracil is unique for RNA. The phosphate functional groups. So this phosphate functional group is derived from phosphoric acid H3PO4. Has 3 active OH groups of which 2 are involved in strand formation. That means DNA formation. The phosphate functional group gives DNA and RNA the property of an acid. So what is an acid? A substance that releases hydrogen ion. Base means a substance that accepts hydrogen ion is a base, and a substance that releases the hydrogen ion is an acid. So at a physiological pH, hence the name nucleic acid. The bonds that are formed from phosphates are esters. So in phosphate, ester bonds are formed. The oxygen atom of the phosphate group is negatively charged after the formation of phosphodiester bond. This negatively charged phosphate ensures the retention of nucleic acid within the cell or nuclear membrane. So these are the 3 subunits of a nucleotide. So what is a nucleotide and nucleoside? The nitrogenous base is a chemically linked to one molecule of sugar, that means at the one carbon of sugar, forming a nucleoside. When a phosphate group is attached to 5 carbons of the same sugar, the nucleoside will become nucleotide. The nucleotides are joined by condensation reaction to form polynucleotide chain. The hydroxyl group on the 3' carbon of a sugar of one nucleotide forms an ester bond with the phosphate of another nucleotide. So the chemical bonds that link the sugar components of adjacent nucleotide are called phosphodiester bond. That means from 5' to 3', indicating the polarity of the strand. The ends of the DNA and RNA are distinct or different. The two ends are designated by the symbols 5' and 3'. The symbol 5' refers to the carbon in the sugar to which the phosphate functional group is attached. The symbol 3' refers to carbon in the sugar to which hydroxyl functional group is attached. So 5' means phosphate group is attached. 3' means hydroxyl group is attached. In RNA, every nucleotide residue has an additional OH group at 2' position at the ribose. Understanding the 5' to 3' direction of a nucleic acid is critical for understanding the aspects of replication and transcription. So based on the X-ray diffraction analysis of Wilkins and Rosalind Franklin, the double helix model of DNA was proposed by James Watson and Francis Crick in the year 1953. The highlight was the base pairing between the two strands of the nucleotide chain. This proposition was based on the observation of Edwin Charles that adenine always pairs with thymine with a double bond with the 2 hydrogen bonds, and guanine pairs with cytosine with 3 hydrogen bonds. The ratio between adenine with thymine and guanine with cytosine are constant and equal. The base pairing confers a unique property to the polynucleotide chain. They are said to be complementary to each other, that is, if the sequence of bases in one strand, that is the template, is known, then the sequence in the other strand can be predicted. For example, in one strand it is adenine, the pairing is thymine. If it has guanine, the pair will be cytosine. So we can easily identify the other sequence. Next, we move on to RNA world. A typical cell contains about 10 times as much RNA as DNA. The high content is mainly due to the variety of roles played by RNA in the cell. 3 molecular biologists in the early 1980s, Leslie Orgel, Francis Crick, and Ozo, these proposed a theory that RNA served as the first stage in the evolution of life on Earth. This hypothesis was called RNA world. Franklin Cornett and Singer in 1957 first demonstrated that RNA is the genetic material in RNA containing virus, that is tobacco mosaic virus. They separated RNA from the protein of RNA viruses. RNA as the genetic material. Now we will see how RNA as a genetic material. The ability of RNA. RNA has the ability to act as both genetic material and catalyst. What is meant by catalyst? Catalyst means an enzyme which helps to speed up the reaction. So catalyst is an enzyme that speeds up the reaction. So in living organisms, several biochemical reactions are catalyzed by RNA. The catalytic RNA is known as ribozyme. Next we see what is ribozyme. Ribozyme means ribonucleic acid enzyme. It is a catalytic RNA. Ribozymes are RNA molecules that have the ability to catalyze specific biochemical reactions. Example, RNA splicing in gene expression. We see what is splicing. Take this picture as an mRNA. This mRNA has two strands. One is the intron, another one is the extron. Introns and extrons are connected or added with help of the ribozyme. This process is called as splicing. Next, the other ability of RNA is some RNA molecules function as gene regulators. They bind to DNA molecules and affect gene expression. So, DNA is a double-stranded molecule having complementary strand, and it is resisted changes by evolving a process of repair. So this can be cued as by this RNA. RNA is a genetic material in some viruses like tobacco mosaic viruses, bacteriophage, theta B, and coronaviruses. So, according to Andrew Fire and Craig Mello, the Nobel Prize winners in 2006, RNA is an active ingredient in the chemistry of life. To summarize, RNA's ability to act as both genetic material and catalyst. It acts as both genetic material and catalyst. RNA functions as a gene regulators, and finally RNA is an active ingredient in the chemistry of life. So these are the ability of RNA. Dear students, I hope you all have understand the concept of molecular genetics. If you have any doubts or if you need any clarification, feel free to ask me. Stay safe at home. Stay strong. Thank you.